In this unit of ANP 202, we'll be taking a look at blood, its characteristics, functions, hematocrit, plasma, the formed elements, including red blood cells, white blood cells, and platelets, as well as hemostasis and blood typing. Some of the characteristics of blood, we see that its color depends on its oxygen content. So a bright red hue is going to be due to more oxygen, less carbon dioxide, and a bluish red hue is going to be due to less oxygen and more carbon dioxide. Blood is a very specialized type of connective tissue. It's a liquid matrix. It's viscous or thick and sticky. Its temperature is about 37 degrees Celsius or 98.6 degrees Fahrenheit, and its pH is slightly alkaline. The volume that an average adult has within their bodies at any point in time is around 5 liters. Some of the functions of blood, we see that it's important for transport, so it's transporting blood gases, oxygen and carbon dioxide between lungs and tissues, also transporting nutrients, hormones, enzymes, antibodies, and other wastes throughout the body. It regulates acid-base balance, or pH, of interstitial fluid. So interstitial, we're in between the cells. And it helps with body temperature because water holds heat, the blood holds heat, and then it also helps with regulation of electrolytes. Blood also helps protect against blood loss through clotting, and it also assists with toxins and disease protection through immunity. Some of the major components of blood, what we see is that plasma is about 55% of whole blood, and it's the least dense component. Then there's a buffy coat in this thin white line that's in between the plasma and the erythrocytes, and the buffy coat is comprised of leukocytes and platelets. It's less than 1% of whole blood, and erythrocytes then are 45% of whole blood. They're the most dense component. When we see erythro, we want to think of the Greek word for red. So these are red cells. And leuko is Greek for white. So leukocytes are going to be the white cells, white blood cells. The formed elements plus plasma, if we look at this then, plasma is the fluid component of blood. And the formed elements that are, they're called formed because they're not considered fluid. The formed elements there are red blood cells or erythrocytes, white blood cells or leukocytes, and platelets or thrombocytes. So thrombo, we'll take a look at some hormones that are released that have to do with coagulation. That makes sense that platelets would be referred to as thrombocytes when we take a look at that. So again, plasma, 55% of blood volume, the fluid portion of blood. It's about 92% water, which then as a solvent, it holds heat. And the solutes that are within plasma make up about 8% of the plasma composition. These solutes are proteins, and albumin, which is a protein, is 60% of the total protein mass within plasma. It's made in the liver, it's responsible for blood viscosity, it creates osmotic pressure in plasma, and it regulates blood volume and blood pressure by attracting water from the tissues. So one of the things that we have to take a look at within this section is how do we address the idea of osmolarity, or the movement of water across a semi-permeable membrane. One of the things that we will see here in this semi-permeable membrane is that semi-permeable means that it is permeable to some components. So there is passage of, say, water and ions that can occur just based on regular diffusion. So if we were to say, I've got 3 of these guys out here, I've got 3 of these blue squares, and 3 of these blue squares. Then just through the nature of diffusion through a semi-permeable membrane, these would pass, and so in an enclosed environment out here in the interstitial fluid, we then would hit an equality of components inside and out of the cell. So just through leak channels and basic diffusion, this is what would happen if there were no other components involved. So water is allowed to move passively, but other components within the mixture may not be, largely due in part because of their size. So if I have a molecule that is like this, these molecules are not going to be able to pass out of the cell. And what will happen then is, as if these cells are elephants in a room, there's less room for the other components that are in the cell. And what will happen then is this will create a demand to still equalize what's outside of the cell in the interstitial fluid and what's inside. So there'll be a progressive move of water into the cell to go ahead and balance what's outside of the cell with what's inside. This is called osmolarity. So one of the major proteins that we see inside plasma, to the tune of about, like we were saying, 65% of all protein that's within the plasma, albumin, is an important carrier protein that we were taking a look at in our endocrine section that will not be able to pass through this semi-permeable membrane. So what this membrane is permeable to is not permeable to large molecules like albumin. Therein lies the semi-permeable name. And so we can have the passage of water out of and into the cell, but we cannot pass these larger proteins, and this creates a much higher osmotic pressure inside, say, this capillary. And so the pressure from outside of the capillary is going to desire to equalize that by sending more water in. So water will move to the side of the plasma membrane that has the greatest osmotic pressure. The difference in osmotic pressure is due to these large proteins that are floating in the bloodstream in the plasma that are not existing out here in the interstitial fluid. And with a semi-permeable membrane, the only thing that can change sides to go ahead and try to create a more equalized pressure between these two spaces is water. Water will move into the location where it is lower in concentration so that it can equalize with the location that it came from. Question of, can osmotic pressure differences affect the shape or volume of a cell? When we ask ourselves the question, can osmotic pressure differences affect the shape or volume of a cell? Along these lines, if we take a look at 3 different scenarios, one of pure water, one of a 0.9% sodium chloride solution or saline, and then another of a 9% sodium chloride solution. First of all, what we would want to ask ourselves, as far as tonicity goes related to osmotic pressure, compared to the tonicity of the blood that this fluid would be intravenously applied to, how do these different solutions relate? If I was to ask you which of these is isotonic, which of these is hypertonic, and which of these is hypotonic, see if you can match these to the correct location. So take a minute and pause the video and try that. If you said that pure water is hypotonic, then you're on track. Hypo is below tonicity. This is going to be a lower osmotic pressure infusion. And as we know, when we enter a lower osmotic pressure item, it has less solute. The cells have a fair amount of solute within them, the water that has been infused from this pure water solution will rapidly enter the cells and cause them to lyse or burst. 0.9% sodium chloride or saline solution, if you said isotonic, or when it's infused into the bloodstream, it has a negligible effect on cells in terms of lysing or crenating the cell. You're correct. That leaves us then with a hypertonic solution of 9% sodium chloride, which here we have a very high osmotic pressure. We have a lot of solute that's coming into this mixture. So if we put this 9% into the bloodstream, and you have a lot of solute in that, water is going to want to follow salt to balance out the pressure. And so the water that's within the cell will pull out into its surrounding fluid, into the surrounding plasma, and these cells will then crenate. The relationship of what type of fluid we put into the bloodstream, the osmotic pressure that exists in interstitial fluid and inside capillary beds all has a very important relationship in terms of the health of the cells in the bloodstream and within the body. We see globulins that make up 33% of total protein. These are going to be antibodies related to immunity, fibrinogen and prothrombin related to the assist within the creation of clots, and these comprise 7% of total protein. Then in serum, this would equal plasma minus clotting factors and proteins. Other dissolved solutes would be amino acids, glucose, fatty acids from digested food, enzymes and hormones, as well as dissolved blood gases that we look at as oxygen and carbon dioxide, electrolytes, sodium, potassium, chloride, bicarbonate, calcium, magnesium, other electrolytes. Electrolytes, in many cases, if you hear of Gatorade or any of these other electrolyte supplements, they're bringing in these components, these elements, into the bloodstream in one fashion or another to try to increase blood volume. The formed elements of blood would be the red blood cells, and the sites of production for red blood cells in the fetus are the liver, spleen, and bone marrow. And in the adult, we take a look more at the red bone marrow, specifically of the ribs, pelvis, sternum, vertebrae, and clavicles. The structure of mature circulating red blood cells are these bioconcave discs. So they're concave on both sides, bioconcave. This gives the red blood cells the greatest surface area to allow them to be available for diffusion of blood gases. The plasma membrane then on these red blood cells bears proteins that make up blood group antigens or blood types. We're going to see these antigens that are on the plasma membrane that help our immune system determine if these cells should be here or not. The plasma membrane is negatively charged, which prevents agglutination of red blood cells. And agglutination just means to gather or form into a mass. The cytoplasm of red blood cells has no nucleus, no Golgi apparatus, no endoplasmic reticulum, no ribosomes, and no mitochondria. All of these are ejected when the cell is created. All of this is so that the red blood cells have more surface area to carry the gases that we need them to carry. Some images then, artwork of these electron micrographs of scans of the cells that are just interesting to see, really pretty when they've been colorized. Which brings us then to erythropoiesis, the genesis of red blood cells. And what we're interested in here is that we have stem cells or hemocytoblasts that then move forward into a committed cell type. So this cell will definitely be one cell type or another. So here we're looking at a proerythroblast that then enters a developmental pathway where we see ribosome synthesis and hemoglobin accumulation, so pulling in these heme units and the hemoglobin, then ejecting the nucleus and becoming a full-fledged erythrocyte. And all of this is stimulated to occur by erythropoietin. When we take a look at the structure on the red blood cells that is actually helping with the transport of gases, we drill into a hemoglobin. The hemoglobin molecule has 4 globin chains, which are protein, and these bind carbon dioxide. Then there are also 4 heme units, or red pigment, that are made of iron, and these atoms bind oxygen. Heme is a red pigment which binds blood gases, and we have about 280 million hemoglobin molecules per red blood cell. The function then of these hemoglobin molecules is to transport blood gases between the lungs and tissues. And some of the other components that we see in the mix, we'll take a look at oxyhemoglobin, deoxyhemoglobin, and carbaminohemoglobin, depending on what state the hemoglobin is in terms of the gases that it's carrying. Hemoglobin then is we have a beta globin chain and an alpha globin chain, just protein structures that are very specific to hemoglobin. And then these heme groups that you'll see an iron ion in the center of these other chains of molecules that form the heme group. And so these heme groups are part of these, tied in with these globin proteins, and together we have hemoglobin. A question comes up then as to why carbon monoxide is so toxic. And the answer is that hemoglobin has a greater affinity for carbon monoxide than for oxygen. And carbon monoxide will actually compete with and displace oxygen from hemoglobin, which reduces the amount of oxygen available to the tissues. Once red blood cells have lived their life, they will be recycled. And part of this process then is that they're engulfed by macrophages in the liver, spleen, and bone marrow, and hemoglobin is broken down. Hemoglobin will be split into its heme and globin units. The globin units will go back into amino acids in the bloodstream. The other cells will go into the formation within the liver of bilirubin. And then iron will be stored in the liver as ferritin, hemosiderin. And iron then is bound to transferrin and released to the blood from the liver as it's needed for erythropoiesis. Bilirubin is picked up from the blood by the liver, secreted into the intestine in bile, and metabolized to stercobilin by bacteria and excreted in feces, giving the feces their brown color. Then food nutrients, including amino acids, iron, B12, and folic acid, are absorbed from the intestine and enter the blood. We take a look now at leukocytes, and remembering that leuko is Greek for white, and these are white blood cells, whereas previously we were looking at the erythrocytes, or the red blood cells, and both leukocytes and erythrocytes are the formed components of blood, and then plasma would be the more liquid portion of blood. So here the leukocytes, we see that leukocytes are actually considered to be complete cells, unlike erythrocytes. And leukocytes are considered to be complete cells because they have nuclei, Golgi apparatus, endoplasmic reticulum, ribosomes, mitochondria, and lysosomes. So the nuclei, Golgi apparatus, endoplasmic reticulum, ribosomes, we're looking here at some kind of protein synthesis mechanism, energy conversion, and then some kind of lysosome action or enzyme activity that they would provide. As we take a look at white blood cell characteristics, we can see diapedesis as a move out of capillaries and a binding to cell adhesion molecules in the endothelium. We see amoeboid motion or movement like an amoeba, and this is how white blood cells will move in the interstitium. Positive chemotaxis or migration toward damaged tissues and attracted by microbial toxins, fluids released by damaged cells, fluids released by white blood cells. So the idea here that these white blood cells are able to move to locations where they need to be, and they're attracted typically by damage or disease processes that happen in the tissues of the body. Some types of white blood cells. We see classification of granular, which would be neutrophil, eosinophil, and basophil, and agranular of lymphocyte and monocyte, and then we have platelets. So you might say, what is the difference between granular and agranular? And granular literally has these granules that can be seen within the cytoplasm, whereas agranular does not. An easy mnemonic we can use to remember the quantity of white blood cells in the bloodstream is to never let monkeys eat bananas. And what this speaks to is that our neutrophil, lymphocyte, monocyte, eosinophil, and basophil. This is the order if we went from highest percentage of existence in the bloodstream to lowest existence. And in terms of your mnemonic, if you wanted to create a mnemonic to keep granular and agranular together, you might just have to do an indication of which of these would be granular to help you remember that. Hemocytoblasts are the stem cells that we will see within the red bone marrow of our skeletal bone. And hemocytoblasts will have two major pathways to cellular creation, either myeloid stem cells or lymphoid. Myeloid will move all the way through its movements to create eosinophils, basophils, neutrophils, monocytes, and then some of these monocytes will become macrophages, and then we'll take a look at macrophages and their outcome in a little bit. Lymphocytes then will become plasma cells. Some lymphocytes will become plasma cells. Neutrophils, as we start to take a look at these as a group, are 60 to 70% of normal total white blood cell count. They have a 2 to 6 lobe nucleus, and they have their granules that stain lavender, and their function is to destroy bacteria. They can do this within a respiratory burst, where O2 is rapidly metabolized by neutrophils to form bacteria-killing oxidizing chemicals. So there's a superoxide radical, oxygen and hydrogen peroxide, that can rapidly kill bacteria. The half-life of neutrophils in circulation is about 6 hours, and there are 100 billion of these produced per day to maintain normal circulating levels in our bloodstream. Lymphocytes are 20 to 30% of normal total white blood cell count. And they have a large nucleus and a thin rim of cytoplasm. This is one of the ways you can identify them in the microscope. After birth, some of these are formed in the bone marrow, and most, however, are formed in lymph nodes, thymus, and spleen. The types of lymphocytes that we see are B lymphocytes, or they're called B cells, which produce antibodies for humoral immunity, so in the bloodstream. And then T lymphocytes, or T cells, are going to seek and destroy foreign material directly. So this is cell-mediated immunity. Monocytes are 4 to 8% of normal total white blood cell count, and they have a large palish nucleus and a lot of cytoplasm. They enter blood from bone marrow and circulate for approximately 72 hours, then enter tissues and become macrophages, and they may persist in tissues for up to 3 months. They do not re-enter circulation. Eosinophils are 2 to 4% of normal total white blood cell count. Their nucleus is 2 lobes in general, but not always. Their granules stain red, or eosinophilic. They're abundant in the gastrointestinal tract, the urinary and respiratory tract walls. They kill parasitic worms, which would include flatworms, tapeworms, and flukes, as well as roundworms, which would be pinworms and hookworms. And they may play a role in allergies and asthma in terms of creating somewhat of a inflammatory response to cause asthma. Basophils are less than 1% of normal total white blood cell count. They have an indented nucleus and granules that stain blue. They're basophilic. they're involved in inflammatory reactions, and they release histamines for vasodilation. So if we look at a cluster of cells under the microscope, we can see that the white blood cells are drastically outnumbered by the red blood cells. This has to do with the larger number of erythrocytes or red blood cells that we see, and the smaller quantity of white blood cells. So again, never let monkeys eat bananas. The neutrophils are at 65%, lymphocytes are at 25%. Then we see the monocytes at 6, and we start dropping drastically from there to eosinophils at 3, and basophils at less than 1%. Here, if we identify this, we see multinucleate, so we would think neutrophil. If you want to try to take a moment to identify this cell in the mix of all these red blood cells, we see a large nucleus with a small amount of cytoplasm, indicating a lymphocyte. Here we see almost a dual-lobed nucleus, a monocyte. Another dual-lobed type of nucleus with red staining granules, so a eosinophil. And then finally, this clustered speckled type of cell among all of the other red blood cells as a basophil. Platelets then are cytoplasmic fragments of megakaryocytes. They have no nucleus. They live only about 10 days in circulation, contain granules, and are capable of amoeboid movement, and they function in hemostasis, which is hemo being blood and stasis to stay in place, so clot formation. The granules in platelets contain serotonin, calcium, enzymes, ADP, adenosine diphosphate, platelet-derived growth factor, and thromboxane A2. So these are components that we see come into play in a cascade effect where we have a positive feedback loop that will help us create blood clots. Platelet formation then is stimulated by thrombopoietin from the liver and kidney. So we go back to our stem cell, a hemocytoblast, see a megakaryoblast, promegakaryocyte, megakaryocyte that then breaks off into platelets. And thrombopoietin can be used to increase platelet production in patients with bone marrow failure. So it can be infused into a patient to assist with greater platelet production. Hemophilia, genetic deficiencies in clotting factors. Hemophilia, blood-loving, to have uncontrolled bleeding. This would be what's considered mild hemophilia after an injection into the left buttock. And hemophilia can also cause painful accumulation of blood in the joints. So uncontrolled bleeding with a lack of clotting ability can be a real problem. Here we can see an electron micrograph of a blood vessel wall post-angioplasty. And all of these little blue speckles are platelets that have come in and are hanging out around this angioplasty zone to try and assist with clotting. We see fibrin helping to form this clot, and all of the different items that these cells recruit to form clots, as well as an additional image of that. And so let's go ahead and let's watch a video on blood clotting.
In a first step towards clot formation, platelets are recruited to the site of vessel injury by now exposed molecules of the vessel wall, such as collagen and von Willebrand factor. This factor mediates the linking of platelets to collagen via a specific receptor in the platelet membrane. The resulting change of shape of the platelet from its resting state into the dendritic form indicates activation. The activated platelet in turn releases prothrombotic molecules such as adenosine diphosphate, ADP. By binding to its receptors, ADP induces aggregation. And recruits further platelets to the site. Thromboxane is another important mediator of platelet activation and aggregation. Under its influence, the platelets cross-link with each other. These interlocking mechanisms cause platelet activation to snowball. The clot grows rapidly. Activated platelets also trigger the coagulation cascade and thus the formation of thrombin. Thrombin, in turn, stimulates platelet activation even further, a continuous feedback loop. Additionally, thrombin induces the formation of fibrin for the mesh stabilizing the clot. The self-reinforcing process of platelet activation, crucial in the formation of blood clots, is an obvious therapeutic target in conditions caused by inappropriately triggered blood coagulation.
So that video was focused mostly on hemostasis, so hemo being the blood and stasis to stay in place, or the stoppage of blood loss from damaged blood vessels. The steps in hemostasis then would be vascular spasm, so vascular smooth muscle will contract when injured, and if we remember that arteries have this vascular smooth muscle in them, we see a contraction when they're injured. And then platelet plug formation, platelets will adhere to each other to form the plug. And then we see coagulation or clotting. So we saw that platelet plug formation and coagulation in the previous video. During clotting, we see a fibrin mesh that reinforces the platelet plug. So fibrin is a specialized protein for platelet plug formation or reinforcement. The first step in hemostasis would be that vascular smooth muscle will contract in response to direct injury, chemicals released by endothelial cells, chemicals released by platelets, reflexes associated with pain, and a vigorous spasm can limit blood loss for approximately 25 minutes. What this is saying is that these endothelial cells are the cells that are epithelial cells that are inside our vessels, and platelets can release chemicals that also call for more platelets, and as we saw in that previous video. And then also we can have reflexes associated with pain that would be nervous system response. The platelet plug, step 2 in hemostasis. Platelets are normally not sticky. The endothelium also releases chemicals that prevent stickiness because we want platelets to form plugs when we want them to, but we don't when we don't want them to. The endothelium is an important portion of this equation that says if it's not injured, then it will release chemicals that prevent sticking of platelets. If it is injured, it will inspire sticking. So when the endothelium is damaged, collagen fibers will be exposed, collagen, the most predominant protein in the body, and platelets will cling to collagen. Von Willebrand factor is released by the endothelium, and platelets adhere to collagen partially due to this factor. And Erich Adolf von Willebrand, who lived from 1870 to 1949, discovered the von Willebrand factor in a Finnish group of people who were lacking this factor and had bleeding problems. The platelet plug is also inspired by ADP, adenosine diphosphate, making the platelets sticky. Serotonin and thromboxane A2 are released by platelets, which increases vascular spasm and the platelet plug. And the plug is produced within 1 minute, but this is temporary. So a really cool image of clotting where we see all these erythrocytes held up by the fibrin and mesh, white blood cells in the mix. Clotting then requires clotting factors, calcium, and vitamin K, and it's usually complete within 3 to 6 minutes. So you'll notice when you cut yourself, your bleeding should be diminishing within a minute of injury. Hemophilia then, the love of blood, is classified as genetic deficiencies in clotting factors that cause uncontrolled bleeding. So if we can't clot, we won't have hemostasis, we'll have hemophilia. Here in these two different pathways that we see to prothrombin activator, what we're looking at here are just pathways that are the steps that look at the intrinsic and the extrinsic pathways in terms of how blood clots are formed. What we see is that we have clotting of blood inside a blood vessel or outside the body. The endothelium will rupture and expose collagen. Then PF3 is released by these platelets that come together, and PF3 is just platelet thromboplastic factor. This is just inspiring more platelets to come gather. There's a cascade of factors that moves up to a point where they're inspired to then activate prothrombin. So once we're at active factor 10, then prothrombin is activated. From an extrinsic pathway or clotting of blood outside the bloodstream, so say damage to cells that don't directly impact a vessel, we would see that blood will escape to the interstitium or the fluid around the cells, and this blood is exposed to tissue factor, which will create a similar cascade leading to active factor 10 and then prothrombin activator. In phase 2, we take off from the prothrombin activator. Prothrombin activator will activate prothrombin to move to thrombin, and then thrombin in phase 3 will go ahead and activate simultaneously fibrinogen to be converted to fibrin. These fibrin molecules will polymerize or come together to form insoluble fibrin strands, so kind of the cargo net mesh, that then this cross-linked fibrin mesh with a stabilizing factor, active factor 13, will go ahead and play into this sort of cargo net effect that helps capture cells and stop bleeding. Some events following clot formation, we see clot retraction, the contraction of platelet pseudopodia. We see pod or ped, we think about feet. So these are sort of the feet-like structures that have grabbed on, that we saw the platelets go ahead and change their shape to create. So we're going to retract those, and what this is going to do is tighten the clot as they pull together and shrink the ruptured area, which brings damaged ends of vessels back together. The platelets will release PDGF, platelet-derived growth factor, and healing will begin. So this stimulates growth of smooth muscle and fibroblasts. Fibrinolysis, then, is the removal of the clot following repair of the vessel. We need to break that fibrin down so that we quit capturing cells and holding this together in this way. We'll be depending on the normal cells of the tissue or the vessel to keep themselves together. Fibrinolysis, then, tissue plasminogen activator is released by the endothelium in contact with the clot. We see plasminogen being incorporated into the forming clot, and this leads to plasmin, which will then dissolve the fibrin clot. So it's a really fascinating combination of events that goes into the formation of the clot in a positive feedback loop as platelets keep releasing their components to go ahead and keep forming the clot, then ending with a negative feedback loop in these events like fibrinolysis to stop that event and go ahead and quit producing clots so that we don't send off a clot into the bloodstream and cause cell damage or death. This brings us to our discussion of blood groups, and we see that there are 30 blood groups, and these blood groups are based on antigens that exist in plasma membranes of red blood cells. So these antigens can provoke agglutination, and agglutination is the gathering or clustering of other cells. So when bound to antibodies, this is not a good thing. What this is saying is that if we have the wrong antigen in the mix, this can provoke agglutination and cause cells to cluster together and end up causing severe damage and may lead to death. The blood groups that we see are ABO and RH. If we have a mismatch on these groups, it can cause robust agglutination. The ABO blood group, the antigens are A and B, and antibodies are anti-A and anti-B. So if we have an A antigen, we're going to have an anti-B antibody, and if we have a B antigen, so if we would always say we're B blood type, then we have an anti-A antibody. And these are naturally occurring, these antibodies. They appear in plasma with no prior antigen exposure at about 2 months of age. So when considering transfusions, most important considerations are the donor, the antigens on the donor's red blood cells. The recipient's plasma may contain antibodies that destroy donor red blood cells, and then this can block the recipient's blood vessels. That agglutination, the buildup of these blood cells, can lead to kidney damage. Donor antibodies are diluted by the recipient's blood, and transfusions are typically packed red blood cells. From the recipient then, the antibodies in the plasma may destroy donor red blood cells. So this is if we have a mismatch, we can have problems. A universal donor type is type O, has neither A nor B antigens on the red blood cells, and may be given to all 4 ABO blood types. Type O has no antigens on the surface of the red blood cell, therefore recipients' antibodies won't react with donor red blood cells. The type O blood type can only receive blood from type O donation. Universal recipient is type AB, meaning that type AB blood types can receive blood from any of the other blood types. Because it has neither anti-A nor anti-B antibodies in the plasma, and both antigens are recognized as self, may receive all 4 ABO blood types, so universal recipient. Some thoughts on the frequency in the U.S. population of a few ethnicities, then blood group AB, B, A, and O. The red blood cells, the antigens, the red blood cell antigens, also called agglutinogens because they cause agglutination. And then just kind of these illustrations that show what plasma antibodies exist and how those relate to the blood types that can be received. The Rh blood group then is a blood group that was discovered in rhesus monkeys, and there are 50 antigens that are all considered to be an Rh factor. However, C, D, and E are the most common that we take a look at. So the Rh blood group is what would give you your plus or your minus on your blood group, and the plus would mean that you have the Rh factor as an antigen on the plasma membrane of your red blood cell. A negative would mean that you do not have the Rh factor on your red blood cell. And the most common of the C, D, and E Rh blood groups that we focus on is D. There are 50 antigens we could look at. D is the most common to have an impact on our cells. So the D antigen, considered the Rh factor. Just be aware that if you're talking about Rh blood groups, there are a lot of them. We focus on one of them because it seems to have the greatest impact on human health. The antibodies then for the D antigen would be anti-D. They're acquired or produced when an Rh negative individual is exposed to Rh antigens. The antibodies for the D antigen are referred to as anti-D, and these are only acquired or produced when an Rh negative individual who wouldn't have the Rh factor is exposed to Rh antigens. Until an Rh negative person would be exposed to Rh antigens or Rh positive, they would not develop anti-D antibodies. This becomes important in pregnancy. So approximately 85% of the U.S. population is Rh positive. Plasma never contains anti-D antibodies. The Rh negative group is 15% of the U.S. population. Plasma may or may not contain anti-D antibodies. So if the individual is exposed to Rh positive blood or sensitized, then, like I was saying previously, he or she will produce anti-D antibodies. So without that exposure, an Rh negative person would not have anti-D antibodies. The problem with the Rh negative, D negative individual transfused with Rh positive or D positive blood is that on the first transfusion, the individual starts producing anti-D antibodies, which is not an immediate problem. But if transfused again with Rh positive or D positive blood, then those anti-D antibodies will react with the Rh positive, D positive red blood cells, and this will cause agglutination or clumping of the blood cells. This problem shows up again in pregnancy, where we see Rh-D incompatibility. So say we have an Rh negative mother, no anti-D antibodies, who then is with a Rh positive father. This will lead to an Rh positive fetus. And during delivery, the Rh positive red blood cells enter the mother's bloodstream, and the mother will produce anti-D antibodies to the fetal red blood cells. So in the first pregnancy, this may not be a problem, but in successive pregnancies, this is considered a high risk. Erythroblastosis fetalis is the term known for hemolytic disease of the newborn. When the mother has these antibodies to D, then those antibodies will attack the fetus. And what we'll see then is that if the fetus is Rh positive, we'll see agglutination in the fetal red blood cells. The fetal liver is too immature to process ruptured red blood cells, and bilirubin will build up quickly in the fetal blood, leading to jaundice or a yellowed look to the skin, anemia, where the blood lacks enough healthy red blood cells or hemoglobin to function correctly and carry oxygen, which will lead to hypoxia. Prevention of this condition will come from an injection of RhoGAM, which is a serum that contains anti-Rh, anti-D antibodies. And within 72 hours of delivering the first baby and or during pregnancy is when this needs to be injected. And this leads to the destruction of the fetal Rh positive, D positive red blood cells that have passed from or to the maternal circulation. And that will allow for the mother to have successive babies potentially without causing a antibody response to the baby's blood. Let's go ahead and watch a video now on blood typing to sort of tie this all together and wrap up our discussion on the blood.
It's often said that despite humanity's many conflicts, we all bleed the same blood. It's a nice thought, but not quite accurate. In fact, our blood comes in a few different varieties. Our red blood cells contain a protein called hemoglobin that binds to oxygen, allowing the cells to transport it throughout the body. But they also have another kind of complex protein on the outside of the cell membrane. These proteins, known as antigens, communicate with white blood cells, immune cells that protect against infection. Antigens serve as identifying markers, allowing the immune system to recognize your body's own cells without attacking them as foreign bodies. The two main kinds of antigens, A and B, determine your blood type. But how do we get 4 blood types from only 2 antigens? Well, the antigens are coded for by 3 different alleles, varieties of a particular gene. While the A and B alleles code for A and B antigens, the O allele codes for neither. And because we inherit one copy of each gene from each parent, every individual has 2 alleles determining blood type. When these happen to be different, one overrides the other, depending on their relative dominance. For blood types, the A and B alleles are both dominant, while O is recessive. So A and A gives you type A blood, while B and B gives you type B. If you inherit one of each, the resulting co-dominance will produce both A and B antigens, which is type AB. The O allele is recessive, so either of the others will override it when they're paired, resulting in either type A or type B. But if you happen to inherit 2 O's, instructions will be expressed that make blood cells without the A or the B antigen. Because of these interactions, knowing both parents' blood types lets us predict the relative probability of their children's blood types. Why do blood types matter? For blood transfusions, finding the correct one is a matter of life and death. If someone with type A blood is given type B blood, or vice versa, their antibodies will reject the foreign antigens and attack them, potentially causing the transfused blood to clot. But because people with type AB blood produce both A and B antigens, they don't make antibodies against them, so they will recognize either as safe, making them universal recipients. On the other hand, people with blood type O do not produce either antigen, which makes them universal donors, but will cause their immune system to make antibodies that reject any other blood type. Unfortunately, matching donors and recipients is a bit more complicated due to additional antigen systems, particularly the Rh factor, named after the rhesus monkeys in which it was first isolated. Rh plus or Rh minus refers to the presence or absence of the D antigen of the Rh blood group system. And in addition to impeding some blood transfusions, it can cause severe complications in pregnancy. If an Rh-negative mother is carrying an Rh-positive child, her body will produce Rh antibodies that may cross the placenta and attack the fetus, a condition known as hemolytic disease of the newborn. Some cultures believe blood type to be associated with personality, though this is not supported by science. And though the proportions of different blood types vary between human populations, scientists aren't sure why they evolved, perhaps as protection against blood-borne diseases, or due to random genetic drift. Finally, different species have different sets of antigens. In fact, the 4 main blood types shared by us apes seem paltry in comparison to the 13 types found in dogs.